Beneath your feet, in the quiet darkness of the forest floor, a conversation is always unfolding, not in sound, not in movement, but in slow, invisible whispers of chemistry and root-bound signals. Trees, ancient, patient, and still, are not as silent as they seem. They warn each other of danger, share nutrients with the weak, and remember seasons long past through networks hidden in the soil. Tonight, we wander slowly into that hidden world, where forests breathe together, think together, and speak in ways humans are only beginning to understand. And if calm, mysterious journeys through the secret lives of nature help you relax or drift towards sleep, consider subscribing, and let the forest's quiet language carry you gently into the night. Forests have always given humans the impression of silence. When we step beneath a canopy of tall trees, the first sensation is often the quiet, a muffled, softened stillness that feels different from any other landscape. Wind dulls as it passes through layered leaves. Footsteps disappear into moss and soil. Even animal sounds feel distant, absorbed by bark, branch, and earth. To human senses, the forest appears calm, passive, almost asleep. Yet this perception is an illusion born from the limits of our biology. Human hearing is tuned for survival in open environments, for detecting predators, speech, sudden movement. We listen for cracks of twigs, wing beats, growls, flowing water. But the forest does not rely on sound as its primary language. Its conversations happen in mediums we rarely perceive: chemical gradients drifting through soil, electrical fluctuations moving across root membranes, microscopic fungal filaments carrying encoded exchanges from one organism to another. What feels like silence to us is, in truth, density, a layered field of communication unfolding too slowly and too quietly for human senses to grasp. Beneath every step lies an underground architecture more complex than most cities. Roots extend outward not randomly, but strategically, probing for water, minerals, and the presence of neighbors. Some roots intertwine, others avoid contact. Many become wrapped in fine fungal threads so thin they resemble silk under magnification. These threads, called mycorrhizae, form symbiotic partnerships with trees, merging plant and fungus into a shared biological system. Through these connections, substances begin to move. Sugars produced in leaves during photosynthesis travel downward into roots. From there, some are transferred into fungal networks, not lost, but traded. In return, fungi deliver minerals extracted from soil particles too small or too distant for roots to access alone. Phosphorus, nitrogen, zinc. Nutrients flow along these living threads like cargo along rail lines. But nutrition is only part of the exchange. Embedded within these flows are signals, molecular messages shaped by stress, abundance, injury, or environmental change. When a tree experiences drought, its internal chemistry shifts. Hormones alter. Root exudates, chemical secretions change composition. These altered compounds seep into surrounding soil and fungal pathways, where neighboring organisms detect them. Receiving trees do not hear in the human sense. Instead, receptors along their roots register chemical concentrations. Cellular processes adjust in response. Stomata may close earlier to conserve water. Growth may slow, defensive compounds may increase. Preparation begins, not because the receiving tree sensed the drought directly, but because another tree signaled its experience first. Communication here is measured not in seconds, but in gradients and accumulation. A signal released in one root zone may take hours or days to influence another tree meters away. Yet forests operate on timescales where such delays remain effective. Seasons unfold slowly. Insect populations build gradually. Drought deepens over weeks. The pace of communication matches the pace of threat. One of the most studied examples of this hidden exchange occurs during insect attack. When leaf-eating insects begin feeding on a tree, physical damage alone does not define the event. Within minutes, the injured leaves release volatile organic compounds into the air, scents imperceptible to humans but meaningful to nearby plants. Simultaneously, chemical distress signals move downward into the roots and outward through fungal connections. Neighboring trees, receiving these cues, begin altering their own leaf chemistry before insects reach them. They may produce bitter tannins, toxic alkaloids, or digestibility-reducing enzymes. By the time insects spread, the forest has already shifted its defenses, a coordinated response emerging without centralized control. What appears above ground as random survival is, below ground, partially collaborative awareness. Electrical signaling adds another layer to this subterranean dialogue. Though trees lack nerves, their cells maintain voltage gradients across membranes, tiny electrical differences essential for nutrient transport and cellular regulation. Under stress, these voltages fluctuate. Research has shown that electrical impulses can travel along root systems and even across grafted root junctions between trees. These impulses move far slower than animal nerve signals, but speed is relative to need. A slow electrical wave passing through roots can still initiate chemical responses ahead of physical threats. It is less like a shouted warning and more like a slowly spreading ripple in water, subtle but consequential. Fungi often mediate these exchanges, acting as living bridges. A single fungal network may connect dozens of trees across species lines. Under microscopes, these networks resemble woven lace branching, fusing, dissolving, and reforming continuously. Through them pass not only nutrients but signaling molecules shaped by evolutionary cooperation. Fungi benefit from this arrangement. Trees feed them carbohydrates. In return, fungi expand the communication reach of forests exponentially. A tree connected to fungal partners gains awareness far beyond the physical limits of its own roots. In dense forests, this creates overlapping webs, networks within networks, where information and resources circulate in patterns scientists are still struggling to map fully. Some trees occupy more central positions within these systems. Older, larger trees possess expansive root zones and long-established fungal partnerships. Because of this, they often function as hubs, receiving, integrating, and redistributing flows across the network. When saplings grow in deep shade where photosynthesis is limited, these older trees may transfer carbon compounds to them through fungal pathways. This transfer is not charity in a human moral sense, but it reveals a system where survival is partially collective. Supporting younger trees maintains forest density, stabilizes soil, and preserves fungal partners shared by all. The illusion of competition dissolves into a spectrum where cooperation and rivalry coexist. Even tree stumps, long cut from their trunks, sometimes remain biologically active underground. Sustained by root connections to living neighbors, they continue exchanging nutrients and maintaining cellular integrity decades after felling. To human eyes, the stump is dead. Underground, it remains integrated into the living network. Silence above does not mean stillness below. Chemical signaling also encodes memory. When trees experience repeated stress, drought cycles, pest invasions, temperature extremes, their cellular machinery adapts. Gene expression shifts. Defensive pathways become more readily activated. Some of these adaptations persist season to season, allowing faster responses to familiar threats. Signals reflecting these experiences can move through root and fungal pathways, influencing neighboring trees' readiness as well. In this way, forests accumulate distributed memory, not stored in a brain, but in patterns of chemical priming across organisms. Time functions differently in such systems. Human communication prioritizes immediacy. Forest communication prioritizes persistence. Signals may linger in soil chemistry long after release. Fungal networks may retain altered flow patterns. Root growth may adjust directionally toward or away from previous exchanges. The forest does not forget quickly. Environmental changes further shape this hidden language. During periods of abundance, ample rain, balanced temperatures, nutrient exchanges become more generous. Carbon flows outward from sunlit canopy giants into shaded understories. Growth accelerates collectively. In scarcity, flows tighten. Trees reduce outward transfers. Fungal mediation becomes more selective. The network constricts, prioritizing survival over expansion. Even then, communication does not cease. Stress signals intensify. coordinating dormancy, leaf shedding, or root contraction in response to shared hardship. To walk through such a forest unaware of these processes is to witness only the surface of a vast living system. The soil itself becomes a medium of conversation. Root exudates alter microbial communities surrounding each tree. Bacteria and fungi shift populations based on these secretions, influencing nutrient availability and pathogen resistance. Neighboring trees detect these microbial changes, adjusting their own root chemistry accordingly. Thus, communication extends beyond direct exchange into environmental modification, shaping the very medium through which signals travel. Scientists studying these systems often rely on isotopic tracing to observe exchanges invisible to the naked eye. By introducing carbon isotopes into one tree's leaves, researchers can later detect those isotopes in entirely different trees connected underground. What appears as isolated individuals reveals itself as a shared metabolic field. Still, the language of trees remains profoundly slow. A human conversation may last minutes. A forest conversation may span seasons. Signals released in spring may influence growth patterns in autumn. Nutrient transfers supporting saplings today may shape canopy composition decades later. Patience defines the grammar of this communication. From a sensory perspective, humans remain largely excluded from this dialogue. We cannot hear electrical pulses in roots. We cannot smell most volatile signals released by leaves. We cannot perceive fungal negotiations occurring in darkness centimeters below our feet. Yet technology increasingly reveals what perception cannot. Microelectrodes detect voltage changes along roots. Soil probes analyze chemical gradients. Imaging techniques map fungal filaments threading through earth. Each discovery dissolves another layer of perceived silence. The forest grows louder the more closely we learn to listen. Philosophically, this challenges long-held assumptions about individuality in nature. Trees once understood as solitary competitors now appear embedded within cooperative infrastructures. Survival emerges not solely from strength, but from connectivity. Even death contributes to the system. Fallen trees decompose, releasing stored nutrients back into soil. Fungal networks reroute these resources to living neighbors. Signals associated with decay alter microbial balances, influencing regeneration zones where new saplings emerge. Endings become messages as much as beginnings. Night introduces yet another shift in forest communication rhythms. With photosynthesis paused, carbon flows slow. Root activity redistributes stored resources. Some signaling pathways intensify in darkness, particularly those related to growth regulation and repair. While humans associate night with stillness, underground exchanges continue uninterrupted. Chemical conversations persisting beneath sleeping landscapes. Seasonal transitions layer additional complexity. In autumn, trees withdraw nutrients from leaves before shedding them. Signals coordinate timing to reduce vulnerability to frost. Fungal partners adjust metabolic rates in tandem. Winter dormancy spreads gradually through signaling cascades rather than instantaneous shutdown. Spring reverses the flow, hormonal cues rising from roots trigger bud formation above. The forest awakens collectively, though no single tree commands the change. All of this unfolds continuously beneath the threshold of human awareness. To stand in a forest clearing at dawn, hearing only wind and distant birds, is to witness merely the audible fraction of a far greater system. Beneath the soil, trillions of microscopic exchanges occur. Resources traded, warnings issued, alliances maintained. Silence in this context is not absence, it is translation failure, a mismatch between human sensory design and ecological communication modes. As research deepens, the metaphor of forests as communities strengthens. Not communities in the human social sense, but integrated living matrices where boundaries blur and functions distribute. The illusion of stillness fades. Trees do not speak in words or sounds, yet they participate in dialogues as real as any spoken language, structured by chemistry, paced by seasons, mediated by fungi, and sustained by shared survival. And so the next time one walks beneath towering trunks, feeling the hush of filtered light and softened air, it may be worth remembering that the quiet is only surface deep. Below, in darkness rich with roots and threads, the forest is speaking endlessly in pulses, in molecules, in patient signals traveling slowly from one life to another. A vast conversation unfolding in a language older than sound and far more enduring than silence. Long before humans imagined cables stretched across oceans or signals traveling invisibly through the air, forests had already built their own vast communication infrastructure beneath the soil. To the casual observer, trees appear as solitary beings. Each trunk rises from the ground as if independent, competing for sunlight, water, and space. Their separation seems obvious: physical gaps between canopies, distinct root systems hidden below. For centuries, this visual distance shaped human assumptions about how forests functioned, individual organisms locked in quiet competition. But beneath the surface, the reality is far more intertwined. Hidden in the darkness of the soil lies an immense biological network formed not by the trees alone, but by their ancient fungal partners. This system, now often called the Wood Wide Web, is composed of mycorrhizal fungi that colonize and connect tree roots through microscopic filaments known as hyphae. These hyphae are astonishing in scale despite their size. Each filament is thinner than a human hair, yet capable of extending meters through soil, branching repeatedly, weaving around stones, penetrating microscopic pores, and linking root to root across surprising distances. Multiply this by billions of filaments operating simultaneously, and the forest floor becomes threaded with living conduits resembling an organic circuitry. Unlike roots, which primarily serve the needs of individual trees, fungal hyphae operate as shared pathways. When fungi attach to a tree's roots, they form a symbiotic interface. The fungus penetrates root tissue or wraps around its exterior, creating exchange structures where nutrients and carbon compounds pass between organisms. The tree supplies sugars produced through photosynthesis, energy-rich molecules fungi cannot produce on their own. In return, the fungus provides minerals and water gathered from soil zones beyond the reach of roots. This partnership alone would be remarkable. Yet the system extends further, because one fungal network can attach to multiple trees at once. It effectively links them into a shared metabolic web. Carbon fixed in the leaves of one tree may travel down into its roots, transfer into fungal tissue, and then move outward to entirely different trees connected along the same network. The analogy to internet cables emerges from this capacity for transmission. Just as digital cables carry packets of information, fungal networks carry packets of chemistry, sugars, nitrogen compounds, stress hormones, defensive signals. Flow rates differ, of course, moving far slower than electronic data, but the principle of connectivity remains strikingly similar. Through this underground web, trees participate in exchanges that reshape how forests function. One of the most profound aspects of this system is resource redistribution. Sunlight is unevenly distributed in forests. Towering canopy trees receive abundant energy, while saplings in deep shade struggle to photosynthesize enough carbon to survive. Yet studies using isotopic tracers have revealed that larger trees often transfer carbon to smaller ones via fungal pathways. These transfers are not random leaks. They follow concentration gradients, physiological demands, and seasonal cycles. During periods when a mature tree produces excess carbohydrates, some of that energy enters the network. Shaded seedlings connected to the same fungal web may then receive supplemental carbon, sustaining their growth until they reach light. This phenomenon challenges the long-standing belief that forests are purely competitive arenas. Instead, they operate partially as cooperative systems where resource sharing enhances collective stability. The fungal network acts as mediator, transporter, and regulator simultaneously. Hyphae possess membranes capable of selective absorption and release. They do not simply allow unrestricted flow. Instead, exchanges occur through controlled biochemical gateways influenced by fungal metabolism, tree demand, and environmental conditions. In this sense, fungi resemble traffic controllers within the network, directing flows where needed, slowing them where scarce, and maintaining overall system balance. Signal transmission adds another layer of complexity. When a tree undergoes stress, insect attack, drought, disease, its internal chemistry changes. Hormones such as jasmonic acid or salicylic acid increase, triggering defensive pathways. Some of these signaling molecules move into root tissues and enter fungal conduits. Neighboring trees connected through the same mycorrhizal web detect these compounds and begin preparing defenses before the threat physically reaches them. Leaves may thicken, bitter chemicals may accumulate, growth may pause to conserve energy. The network thus becomes an early warning system, not conscious, but responsive. Speed is relative to ecological context. Those signals may take hours or days to propagate. This timescale aligns with the spread of many environmental threats. Insects move gradually. Soil pathogens expand slowly. Drought intensifies over weeks. Early signaling still provides meaningful adaptive advantage. The structure of the wood wide web varies by forest type. Some fungi form ectomycorrhizal associations, wrapping around roots externally. Others penetrate root cells internally, forming arbuscular networks resembling microscopic trees within roots themselves. Different fungal species specialize in different host trees, creating overlapping but distinct subnetworks within the same soil. This layered connectivity produces resilience. If one fungal species declines due to environmental stress, others may continue linking trees, preserving communication routes. Old-growth forests often display the densest network architectures. Decades or centuries of uninterrupted fungal expansion create deeply integrated systems where nearly every tree is connected, directly or indirectly. Large, ancient trees function as central nodes due to their extensive root systems and long-standing fungal partnerships. These hub trees, sometimes called mother trees, play disproportionate roles in network dynamics. Their carbon output sustains numerous fungal partners, which in turn connect them to wide clusters of younger trees. Through this position, they distribute resources, stabilize soil ecosystems, and influence regeneration patterns across large areas. Even species boundaries blur within these systems. Birch trees and fir trees, for instance, have been documented exchanging carbon seasonally through shared fungal networks. When birch trees leaf out earlier in spring, they may supply carbon to shaded fir seedlings. Later in the year, when fir trees remain photosynthetically active longer, the flow can reverse. Reciprocity emerges not from intention, but from physiological timing mediated by fungal pathways. The soil becomes less a battlefield and more a marketplace. Exchanges governed by availability, demand, and mutual survival. Fungal networks also extend communication beyond trees. Shrubs, grasses, and even some non-photosynthetic plants tap into the wood wide web. Certain orchid species, for example, rely entirely on fungal carbon transfers during early development, effectively parasitizing the network until they can photosynthesize independently. This broadens the web into a multi-species infrastructure where energy and information circulate widely. Yet participation is not always equal. Some plants exploit the network disproportionately, drawing more carbon than they contribute. Fungi may respond by restricting connections or reallocating flows, maintaining systemic balance. Negotiation in biochemical form persists constantly underground. Environmental disruptions can fracture these networks. Logging, soil compaction, pollution, and climate change all affect fungal viability. When networks break, resource sharing declines. Seedling survival rates drop. Forest resilience weakens. This reveals how deeply forest health depends not only on trees themselves, but on the invisible fungal architecture supporting them. Reforestation efforts increasingly recognize this reality. Planting trees without restoring mycorrhizal communities often yields poor results. Saplings grow slower, resist disease less effectively, and struggle to establish root partnerships. Introducing compatible fungal inoculants can dramatically improve forest recovery, essentially rebooting the wood wide web alongside tree planting. The analogy to technological networks deepens here. Just as digital infrastructure requires routers, relays, and maintenance, biological networks rely on fungal diversity, soil chemistry, and ecological continuity. Damage to the network affects the entire system's performance. Still, the Wood Wide Web differs from human internet systems in one profound way. It evolved without centralized design. No organism planned the network. It emerged gradually through millions of years of symbiosis. Fungi that connected multiple trees gained more carbon. Trees that partnered with expansive fungi accessed more nutrients. Mutual reinforcement drove increasing complexity. Evolution, not engineering, built this communication web. Time operates differently within it. Signals travel slowly but persist. Carbon transferred today may be stored in fungal tissues and redistributed months later. Seasonal pulses move through networks like tides, expanding in spring growth, contracting in winter dormancy. The forest breathes through its web. Even decomposition feeds the system. When a tree dies, its roots remain connected for years as fungal partners break down tissues. Nutrients released through decay enter the network and move into living trees nearby. Death becomes redistribution rather than termination. Thus, the Wood Wide Web integrates life cycles, growth, stress, death, regeneration into a continuous exchange system. Technological metaphors help human understanding, but they remain imperfect. Unlike cables, fungal hyphae are alive. They grow, retreat, branch, and self-repair. They respond to chemical gradients, seeking nutrient-rich zones or stressed hosts. Network topology shifts dynamically rather than remaining fixed. It is less like a static internet and more like a living neural mesh, adaptive, self-organizing, constantly remodeling. Research continues to uncover new dimensions of this connectivity. Studies explore whether signal strength varies by kinship, whether related trees exchange more resources. Others investigate how climate change alters fungal species composition, potentially reshaping communication efficiency. The deeper science looks, the more intricate the system appears. And yet, despite this complexity, it remains almost entirely invisible to human senses. A hiker may walk for hours through an ancient forest, never realizing that beneath each step lies a communication web linking thousands of organisms in shared exchange. No sound announces it. No movement reveals it. Only careful instruments and patient study uncover its presence. The illusion of separateness dissolves under this knowledge. Forests are not collections of individuals standing side by side. They are integrated systems bound by fungal threads, metabolic, communicative, cooperative infrastructures sustaining ecosystem balance. Through the Wood Wide Web, trees extend awareness beyond their physical bodies. They detect distant stress, share surplus energy, nurture young growth, and recycle the fallen. It is a language written not in words but in molecules, a network older than humanity's first roads, a system operating in darkness, silence, and patience, yet essential to the vitality of the living world above. And so beneath every forest path, beyond the reach of light, the web persists, carrying whispers of chemistry from root to root, sustaining conversations that have unfolded for millions of years, slow, steady, and profoundly alive. In the deep architecture of a forest, not all trees stand equal. To human eyes, age in a forest is often measured by height, trunk width, or the roughness of bark. The tallest trees seem dominant simply because they reach the light first. Yet beneath the soil, where visibility disappears and true influence unfolds, age expresses itself differently. Older trees, those that have stood for centuries, extend far beyond their visible form. Their roots reach outward in vast radial systems, layered over decades of slow expansion. Around these roots gather dense colonies of mycorrhizal fungi, themselves grown thick through long partnership. Together, root and fungus form a structure of connectivity so extensive that these ancient trees begin to function less as individuals and more as centers, biological hubs within the living network of the forest. Scientists studying these systems have come to call such trees mother trees, not as a poetic flourish, but as a functional description of their role. They anchor the underground web. They gather, store, and redistribute resources. They influence the survival patterns of younger growth around them. Their presence shapes not only the present forest, but its future generations. Time is what builds their power. A young tree begins with a modest root system, shallow and limited. Its fungal partnerships are few, newly formed, and localized. Resource intake is narrow. Communication range is small. It survives primarily on what it can gather alone. But as decades pass, roots lengthen, thicken, and branch. Each season adds new fungal connections. Each drought survived strengthens exchange pathways. Each cycle of growth increases stored carbon reserves within trunk and root tissues. Slowly, the tree becomes deeply embedded within the fungal web, connected to dozens, sometimes hundreds of neighboring plants. Through this embeddedness, influence emerges. Carbon, the foundational energy currency of forests, flows abundantly through mature trees. Their expansive canopies capture vast sunlight. Photosynthesis operates at high volume. Sugars produced in leaves travel downward into storage tissues, filling root systems with surplus energy beyond immediate survival needs. Some of this carbon enters fungal networks. From there, it does not remain stationary. Studies using carbon isotopes have shown that mature trees transfer measurable amounts of carbon to surrounding saplings, particularly those growing in shade where photosynthesis is limited. These transfers occur through fungal conduits connecting root systems, microscopic bridges formed over years of symbiosis. To the sapling, this carbon may mean the difference between survival and starvation. Deep beneath the canopy, where sunlight filters thinly, energy is scarce. Leaves produce little. Growth stalls. Yet through connection to a mother tree, supplemental carbon arrives, not rapidly, but steadily enough to sustain metabolism and root development. In this way, the older tree functions as an energy reservoir for the forest's youth. This redistribution is not indiscriminate. Research suggests that mother trees preferentially transfer resources to their own genetic offspring when possible. Through subtle chemical markers in root exudates, trees can differentiate kin from non-kin. Fungal pathways linked to related seedlings may receive stronger carbon flows than those connected to unrelated species. The mechanism is not conscious recognition, but biochemical compatibility, receptor interactions shaped by shared genetic signatures. Yet the outcome resembles familial support, parent trees nourishing their descendants through hidden channels. Such kin-biased transfer increases evolutionary fitness. By supporting genetically related saplings, mother trees help propagate their lineage even after death. Still, support often extends beyond kin. Mother trees also distribute resources to unrelated seedlings and even different species. Forest stability benefits from diversity. Supporting surrounding growth maintains soil structure, fungal viability, and canopy continuity, all of which feed back into the survival of the mother tree itself. Cooperation and self-interest merge into ecological interdependence. Water distribution adds another dimension to this role. In drought conditions, older trees with deep root systems can access groundwater reserves unreachable by shallow, rooted saplings. Through fungal connections, some of this water moves outward, not as bulk flow, but through osmotic gradients that ease dehydration stress in nearby plants. Signals accompanying this water transfer may prime saplings to close stomata earlier, conserving moisture preemptively. Mother trees thus function as hydraulic stabilizers within the forest matrix. Nutrient cycling reinforces this hub dynamic. When leaves fall and decompose around the base of large trees, nutrient concentrations increase locally. Fungi processing this organic matter absorb nitrogen, phosphorus, and micronutrients, routing them through their networks. Because mother trees host extensive fungal colonies, they become redistribution centers for these recycled nutrients. Saplings connected to the network benefit from this mineral circulation, even if rooted in poorer soil patches meters away. The mother tree's reach extends chemically where its roots alone cannot. Electrical signaling also appears amplified in these central trees. Due to their numerous root grafts, physical fusions between roots of neighboring trees, Electrical impulses generated by stress or environmental change can propagate outward more efficiently. A disturbance detected by a mother tree may ripple through grafted roots and fungal channels, alerting surrounding vegetation faster than isolated trees could manage alone. These grafts, formed when roots grow in contact and fuse vascular tissues, create direct physiological bridges, allowing fluid and signal exchange without fungal mediation. Mother trees often possess the highest number of such grafts simply due to age and root spread. Even after death, their influence persists. When a mother tree falls, its trunk may decompose over decades, but underground its root system remains integrated within the network. Fungal partners continue inhabiting decaying tissues, redistributing stored carbon and nutrients outward. Nearby saplings often flourish in the light gap created by the fallen giant, sustained partly by the nutrient pulse released through its decomposition. Thus, the death of a mother tree becomes an act of final provisioning, feeding the next generation both materially and spatially. Forest regeneration patterns frequently trace back to these cycles. Seedlings growing near former mother trees display higher survival rates, faster growth, and stronger fungal connectivity. The legacy of the central tree continues in structural form through the network it helped build. The concept challenges traditional views of plant individuality. Rather than isolated organisms competing blindly, forests resemble extended families, elders sustaining youth, youth eventually replacing elders, all linked through fungal inheritance systems. Mother trees also influence microbial communities. Their root exudates, chemical secretions released into surrounding soil, shape bacterial and fungal populations. These microbes affect nutrient availability, disease resistance, and root growth patterns for nearby plants. Saplings establishing within this conditioned soil environment benefit from microbial assemblages curated by the mother tree's long presence. It is ecological mentorship encoded in chemistry. Light distribution intersects with this underground care. Large trees inevitably shade the forest floor, limiting photosynthesis for seedlings. Without compensatory mechanisms, this shading would suppress regeneration. Carbon transfers through the wood wide web partially offset this disadvantage, allowing saplings to survive long enough to reach canopy openings created by fallen trees. Thus, mother trees both suppress and sustain youth, a balance maintaining forest continuity. Climate stress further highlights their importance. During prolonged droughts or temperature extremes, forests with intact mother tree networks display greater resilience. Resource sharing buffers localized failures. Saplings that might perish in isolation survive through connected support. Remove the mother trees, and this buffering collapses. Clear-cut logging illustrates the consequence starkly. When central trees are removed, fungal networks fragment. Nutrient redistribution declines. Seedling mortality rises. Even replanted forests struggle because the original hub infrastructure, built over centuries, is gone. Reforestation without mother trees often yields simplified, less resilient ecosystems. Some forestry practices now intentionally preserve large, old trees within logged areas to maintain network continuity. These retained elders continue supporting regrowth through underground exchanges, even when surrounding trees are harvested. The recognition of their ecological centrality is reshaping conservation strategies. Communication through mother trees also includes warning signals. If pests infest a central tree, chemical distress compounds enter fungal channels, alerting surrounding vegetation. Saplings receiving these signals increase defensive chemistry, producing toxins, strengthening cell walls, altering leaf palatability. Preparation spreads outward from the hub like ripples from a dropped stone. Fire response offers another example. After low-intensity fires, surviving mother trees often facilitate regeneration by channeling nutrients toward damaged saplings. Fungal networks reroute flows to stressed zones, accelerating recovery. In this way, the elder trees function as recovery coordinators within disturbed landscapes. Temporal perspective deepens appreciation of their role. A mother tree standing today may have begun life before modern human industry existed. Its root system may have connected with generations of fungi, plants, and microbes long vanished. The network it anchors represents accumulated ecological memory, pathways refined by centuries of exchange. Saplings linked to such a tree inherit not just nutrients, but connectivity, immediate access to an established communication grid. The forest past supports its future through these living archives. Recognition of offspring occurs through root chemistry. Studies suggest that seedlings grown from a mother tree's seeds form more robust mycorrhizal connections with that tree compared to unrelated seedlings. Carbon transfers measured between kin pairs often exceed those between strangers. Chemical signaling pathways appear more permeable, resource flows more generous. Evolution favors such selective support because it propagates shared genetic material. Yet forests avoid becoming monocultures because fungal networks remain open systems. Cross-species transfers maintain diversity, preventing dominance by any single lineage. Balance persists through distributed exchange rather than centralized control. Even spatial arrangement reflects mother tree influence. Seed dispersal patterns often cluster offspring near parent trees. These clusters align naturally with the parent's fungal network, granting immediate connectivity advantages. Saplings germinating within this zone plug into the existing web like devices joining an established grid. Survival probability increases dramatically compared to isolated seedlings beyond network reach. Wind animals and terrain may scatter seeds farther, but those landing within the maternal network benefit first. The metaphor of motherhood extends further in the protective, buffering mother trees provide. Dense root systems stabilize soil, reducing erosion. Their canopies regulate microclimates, moderating temperature swings, retaining humidity, shielding saplings from harsh sunlight or frost. Above and below ground, they create environments conducive to growth, and yet, this centrality carries vulnerability. When disease strikes a mother tree, the network that once radiated strength can also transmit pathogens. Fungal conduits, while beneficial, may carry harmful organisms. The same connectivity that supports life can distribute risk. Forests balance this through species diversity and network redundancy, preventing single-point collapse. Still, the loss of a major hub reverberates widely. Remove a mother tree and resource flows reroute awkwardly. Saplings accustomed to its support may falter. Fungal partners must seek new hosts. The structural coherence of the underground web weakens. Recovery takes decades, sometimes centuries. Understanding this has shifted ecological philosophy profoundly. Forests are no longer seen merely as timber reserves or carbon stocks, but as relational systems where age confers functional responsibility. Old trees are not relics of past growth. They are active participants in sustaining present life. Their stillness hides immense influence. To stand beside a massive, ancient tree is to stand beside a living nexus, a being whose reach extends invisibly through soil, whose stored sunlight feeds unseen generations, whose chemical signals ripple outward in slow waves of information. They do not move. They do not speak in sound. Yet through roots, fungi, nutrients, and signals, mother trees nurture forests continuously, guiding survival, shaping growth, and anchoring the vast, patient community beneath the forest floor. In the quiet architecture of a forest, danger rarely arrives without announcement. To human perception, an insect infestation appears sudden, leaves chewed overnight, bark perforated, branches weakened. We imagine attack as an isolated event, one tree suffering while others stand unaware. Yet beneath this visible damage unfolds a communication process as deliberate and coordinated as any alarm system. When insects begin feeding on a tree, the event is not merely physical consumption, it is also chemical disruption. The first bite, the tearing of leaf tissue or the piercing of bark, triggers immediate biochemical reactions within the plant's cells. Damaged membranes release signaling compounds. Enzymes activate. Hormonal cascades surge through vascular tissues. The tree does not feel pain in the human sense. but it registers injury with remarkable sensitivity. Within moments, the attacked area begins producing defensive chemicals. Some of these compounds are toxic or unpalatable to insects: tannins that bind proteins, alkaloids that interfere with digestion, phenolics that damage cellular processes in herbivores. Leaves may become bitter, bark may harden, sap chemistry may shift, yet the response does not remain localized. Chemical signals travel internally first, moving through the tree's vascular system. Hormones such as jasmonic acid and salicylic acid spread from the wounded site to intact leaves and distant roots. This internal messaging prepares unaffected tissues for possible attack, triggering preemptive defense production before insects spread. But the warning extends even further. Damaged leaves release volatile organic compounds into the air, complex scent molecules invisible to human noses but potent in ecological communication. These airborne signals drift outward on currents of wind, dispersing through the forest canopy. Neighboring trees, upon detecting these compounds through stomatal openings in their leaves, initiate defensive responses of their own. Though untouched physically, they begin producing toxins, strengthening cell walls, and altering leaf chemistry in anticipation. The forest responds collectively to individual injury. Airborne signaling functions as the fastest communication route, but it is not the only one. Simultaneously, underground channels activate. Chemical distress compounds travel downward into the roots of the attacked tree. From there, they enter mycorrhizal fungal networks linking multiple trees. Within these living conduits, molecular signals move outward, carried through hyphal filaments that bridge root systems. Trees connected to the same fungal network receive these chemical cues through root receptors. Their internal signaling pathways activate in response, initiating defensive chemistry hours or days before insects physically reach them. The fungal web becomes an underground alarm cable. This dual signaling system, airborne above, fungal below, creates layered communication redundancy. If wind dispersal fails due to still air or dense canopy, fungal transmission continues. If fungal networks are sparse, volatile signals still carry warning. The forest ensures that distress rarely goes unnoticed. The defensive responses triggered by these warnings vary by species and threat type. Some trees increase tannin concentrations in leaves, making them harder to digest. Others produce terpenoids, aromatic compounds toxic to many insects. Certain species release latex, like sap that traps or poisons feeding organisms. Conifers may flood damaged bark with resin, sealing wounds and immobilizing invaders. Timing is critical. Producing defensive chemicals requires energy and resources. Trees cannot maintain high toxin levels constantly without compromising growth. Warning signals allow them to deploy defenses only when risk is imminent, conserving resources while maximizing survival. Preparation replaces panic. In some cases, airborne signals recruit not only plant defenses but animal allies. When caterpillars feed on certain trees, the trees release volatile compounds that attract predatory insects, parasitic wasps, for example, that lay eggs inside caterpillars. The feeding larvae then become hosts to their own predators, reducing herbivore populations. The attacked tree, in effect, calls for biological reinforcements. Neighboring trees detecting these same signals may also release predator, attracting compounds preemptively, creating zones of amplified defense where herbivore survival becomes increasingly difficult. Defense becomes ecological orchestration rather than solitary resistance. Root signaling introduces even more nuance. Studies using isotopic labeling and chemical tracking have shown that distress signals transmitted through fungal networks can trigger gene expression changes in receiving trees. Defensive pathways activate not only chemically but genetically, upregulating enzymes involved in toxin synthesis. This preactivation allows faster, stronger responses once insects arrive. Speed matters in herbivory events. A delay of even a few days can mean the difference between minor damage and widespread defoliation. Early warning increases forest resilience significantly. Interestingly, signal specificity also appears to exist. Different insect species trigger different chemical responses. Leaf-chewing caterpillars produce one profile of distress compounds. Sap-sucking aphids trigger another. Trees detecting these signals tailor their defensive chemistry accordingly, producing toxins most effective against the specific threat. Communication here carries informational depth, not just alarm. The scale of this signaling network expands with forest density. In tightly connected ecosystems, one attacked tree may initiate defensive responses across dozens of neighboring trees. Fungal networks spanning large areas distribute warning compounds widely. Airborne signals drift through overlapping canopies, amplifying reach. An insect outbreak thus encounters not isolated victims, but a chemically fortified community. Even young saplings participate in this exchange. Though smaller and less resource-rich, they still receive and respond to distress cues. Their defensive chemistry may activate at lower intensity, but the preparation increases survival odds. Mother trees often play amplified roles in warning transmission. Due to their extensive fungal connections, distress signals entering their root systems may redistribute more broadly. As central hubs, they function as relay stations, receiving, integrating, and passing warnings deeper into the network. Their position enhances communication efficiency. Environmental conditions influence signaling effectiveness. Wind patterns shape airborne compound dispersal. Humidity affects volatile molecule persistence. Soil composition impacts fungal transmission speed. Seasonal growth stages alter receptor sensitivity in leaves and roots. Yet despite these variables, the signaling system remains remarkably reliable. Even forests separated by species diversity maintain functional warning exchanges. Cross-species detection of volatile compounds allows mixed ecosystems to respond collectively to threats affecting only certain trees. The language of distress transcends taxonomy. Chemical memory also plays a role. Trees previously exposed to herbivore attack often respond more rapidly to subsequent signals. Their defensive pathways remain partially primed, gene expression systems ready for faster activation. This memory may persist across seasons, enhancing long-term resilience. Signal exposure shapes future readiness. Costs accompany these defenses. Producing toxins diverts energy from growth and reproduction. Leaves fortified with tannins photosynthesize less efficiently. Resin production consumes stored carbon reserves. Trees must balance survival against vitality. Warning signals help optimize this balance, ensuring defenses activate only when needed. In this sense, communication preserves both life and efficiency. Human discovery of these processes emerged gradually. Early observations noted that trees downwind of insect infestations showed increased chemical defenses even before damage occurred. Controlled experiments later confirmed volatile compound detection between plants. Further research using mesh barriers demonstrated underground signaling independent of air transmission, implicating fungal networks. Isotopic tracers and molecular assays revealed the pathways more clearly. Science slowly translated the forest's invisible language. The implications extend beyond academic curiosity. Understanding plant warning systems informs pest management strategies. Agricultural practices now explore inducing defensive signaling in crops using synthetic analogs of plant distress compounds. By simulating attack signals, farmers can prime plant defenses without actual infestation. Forests themselves rely on these systems naturally. When bark beetles invade conifer stands, attacked trees release terpene-rich distress volatiles. Neighboring trees respond by increasing resin production, sometimes expelling beetles physically. In large infestations, however, signaling may not outpace insect population growth, leading to widespread mortality. Even so, warning systems slow spread and shape outbreak dynamics. Fire damage introduces similar signaling. Trees exposed to smoke or heat stress release compounds detected by surrounding vegetation. Defensive pathways linked to drought and heat tolerance activate, improving survival during prolonged fire seasons. Communication extends beyond herbivory into environmental stress adaptation. Night alters signaling rhythms slightly. Some volatile emissions decrease without sunlight, While root-based communication persists, fungal networks continue transmitting chemical information regardless of diurnal cycles. The underground web never sleeps. Philosophically, these warning systems reshape perceptions of plant life. Trees once considered passive now reveal proactive defense coordination. Injury triggers messaging. Messaging triggers preparation. Preparation influences survival across community scales. The forest behaves less like a collection of isolated organisms and more like a distributed sensory system, detecting, transmitting, and responding collectively. Still, this communication remains entirely silent to human ears. No sound marks the release of volatile compounds. No visible signal travels fungal threads. The alarm spreads invisibly, encoded in molecules drifting through air and soil. Silence again proves deceptive. A single caterpillar bite may initiate cascades of chemistry affecting trees meters away. A bark beetle borehole may ripple warnings across fungal networks. Threat transforms into information. Information transforms into defense. And defense, coordinated through unseen channels, becomes the forest's quiet shield against the countless small predators that feed upon it. Thus, when one tree suffers, it rarely suffers alone. Its injury becomes message. Its distress becomes signal. Its warning becomes preparation for the many rooted lives surrounding it. A conversation of survival unfolding slowly, chemically and visibly, beneath the stillness of leaves and the illusion of forest silence. In the filtered light beneath a dense forest canopy, survival is never evenly distributed. Sunlight, the fundamental engine of plant life, arrives in fragmented patches. Tall, mature trees absorb the majority of it high above the ground, their leaves forming layered roofs that intercept energy before it can reach the understory. Below, smaller trees and saplings exist in a perpetual twilight, living, but limited. Their leaves produce only modest amounts of sugar through photosynthesis, often barely enough to sustain respiration and slow growth. For much of scientific history, this imbalance was interpreted through the lens of competition. Forests were seen as arenas where the strong thrived and the weak perished. A struggle for light, water, and nutrients governed by ruthless natural selection. But beneath the soil, where visibility fades and roots intertwine, a different story unfolds, one defined not solely by competition, but by exchange. Healthy, well-established trees do not exist in isolation. Their roots are interwoven with networks of mycorrhizal fungi, microscopic filaments that extend far beyond the reach of individual root systems. These fungal threads connect multiple trees simultaneously, forming conduits through which nutrients and biochemical compounds travel. Within these living pathways, resources move, not randomly but directionally. Carbon, produced in the leaves of sunlit canopy trees, travels downward through phloem tissues into roots. From there, a portion of this carbon enters fungal partners in the form of sugars and carbohydrates, payment for mineral acquisition services. Yet fungi do not simply store this energy. They redistribute it. Through hyphal connections linking neighboring trees, carbon flows outward from energy-rich donors to energy-poor recipients. Saplings growing in shade, their photosynthetic capacity limited, receive supplemental carbon that supports cellular respiration, root expansion, and slow but steady growth. This transfer has been demonstrated through isotopic tracing experiments. Scientists introduce carbon isotopes into the leaves of a mature tree and later detect those isotopes in entirely different trees connected underground, even when no physical root contact exists between them. Energy, it seems, is shared. Sugars form the primary carbon currency in these exchanges. Glucose, sucrose, and other photosynthetic products move along fungal gradients from zones of surplus to zones of scarcity. For a shaded sapling, receiving even small quantities of these sugars can mean the difference between dormancy and death. Growth in deep forest understories often depends on this hidden supplementation. Nitrogen sharing adds another dimension to the cooperative system. Nitrogen, essential for amino acids, proteins, and chlorophyll production, is often unevenly distributed in soil. Some trees, through root depth, microbial partnerships, or location access nitrogen more effectively than others. Mycorrhizal fungi absorb nitrogen in organic and inorganic forms, converting it into compounds usable by plants. Through the same fungal conduits that carry carbon, nitrogen can move laterally between trees. A nitrogen-rich tree may indirectly supply nitrogen to neighbors rooted in poorer soil patches. In return, carbon flows may balance the exchange, forming reciprocal trade relationships mediated by fungal brokers. The system resembles a metabolic marketplace, resources moving along gradients shaped by need, availability, and fungal regulation. Phosphorus, potassium, and micronutrients follow similar pathways. Because fungal hyphae penetrate microscopic soil pores inaccessible to roots, they extract minerals otherwise unavailable to trees. Once absorbed, these nutrients enter fungal networks and may be distributed across multiple plant partners. Thus, a tree's nutrient supply is influenced not only by its own roots, but by the combined reach of the fungal web it inhabits. Healthy trees possessing abundant carbon reserves effectively subsidize the nutrient acquisition efforts of the network, fueling fungal expansion that benefits all connected members. The result is an ecosystem where survival is partially communal. Shaded trees, sometimes called suppressed individuals, often persist for decades in low light conditions. Their growth remains minimal, their trunks thin, their leaves sparse, yet they remain alive, sustained by fungal carbon transfers from canopy giants above. When a canopy gap opens, perhaps through stormfall or the death of an old tree, these suppressed saplings are already established. With sudden access to sunlight, they accelerate growth rapidly, filling the ecological vacancy. Without prior nutrient sharing, many would never survive long enough to seize such opportunities. Cooperation thus supports long-term forest regeneration. Seasonal variation influences sharing dynamics. During peak photosynthetic periods, late spring through summer, carbon surpluses increase in mature trees. Transfers through fungal networks intensify, supporting understory growth and microbial activity. In autumn, as leaves senesce and photosynthesis declines, carbon flows contract. Winter dormancy reduces exchanges further, though some underground transfer persists where soil temperatures remain above freezing. The forest nutrient economy pulses with the seasons. Kinship influences distribution patterns as well. Studies suggest that mature trees preferentially allocate more carbon to genetically related saplings when fungal connections allow. Root exudate chemistry helps fungi differentiate between kin and non-kin hosts, subtly shaping resource flows. Yet this preference does not exclude unrelated trees entirely. Cross-species sharing remains widespread. Birch may supply fir, pine may support spruce. Diversity strengthens network resilience, ensuring that resource flow continues even if one species suffers decline. Cooperation operates across genetic and species boundaries. Environmental stress amplifies sharing behavior. During drought, carbon transfers may increase toward water-stressed trees, supporting root maintenance and osmotic regulation. In nutrient-poor soils, mineral redistribution intensifies. After insect outbreaks or disease events, surviving healthy trees channel resources toward recovering neighbors. The network responds dynamically to ecological pressure. Fungal mediation ensures that sharing remains balanced. Hyphae regulate exchange through biochemical gating, allowing transfer where beneficial, restricting it where exploitative. Plants that contribute little carbon may receive reduced nutrient support in return. This prevents systemic drain by parasitic participants. Even within cooperation, equilibrium persists. The cost to donor trees remains measurable but often modest. Transferring carbon and nutrients requires energy investment. Growth rates in donor trees may slow slightly under heavy redistribution demand. Yet the long-term benefits, maintaining forest density, preserving fungal partners, stabilizing soil, often outweigh immediate losses. Survival in forests is rarely solitary. The idea of cooperation challenges classical evolutionary interpretations emphasizing competition alone. While trees certainly compete for light and space, fungal-mediated sharing introduces a parallel dynamic. one where mutual support enhances ecosystem stability. Forests, in this view, resemble interdependent societies more than battlegrounds. Carbon sharing even extends to non-photosynthetic plants. Certain understory species lack chlorophyll entirely, deriving all carbon through fungal intermediaries connected to photosynthetic trees. These plants function as full network dependents, sustained entirely by the metabolic productivity of others. Though sometimes labeled parasitic, their presence contributes to biodiversity and fungal diversity within the ecosystem. Decomposition integrates into this nutrient exchange web. When leaves fall or trees die, fungal decomposers break down organic matter, releasing stored nutrients. These nutrients enter fungal transport pathways, redistributing into living trees. Carbon fixed decades earlier in now fallen trunks returns to circulation, sustaining new growth. Death becomes nutrient donation. Spatial patterns of sharing often reflect fungal network density. Areas near large, mature trees host the most extensive hyphal webs, facilitating stronger nutrient flows. Saplings germinating within these zones gain early access to cooperative support. Those outside network reach face harsher survival conditions. Connectivity shapes fate. Human disturbance disrupts this system profoundly. Clear-cut logging severs fungal networks. Soil compaction damages hyphal pathways. Chemical pollutants alter microbial balance. Without intact fungal infrastructure, nutrient sharing collapses. Replanted forests often struggle precisely because this cooperative web has been destroyed. Modern ecological restoration increasingly includes fungal inoculation, reintroducing mycorrhizal species to reestablish nutrient exchange pathways. Success rates in sapling survival rise dramatically when fungal networks recover. Technology begins to respect ancient biological systems. Even climate regulation ties into nutrient sharing. Carbon stored in forests does not remain locked within individual trees alone. Through fungal redistribution, carbon supports soil microbial communities, influencing decomposition rates and soil carbon sequestration. The wood wide web thus participates in global carbon cycling. Nighttime introduces subtle shifts in sharing dynamics. With photosynthesis halted, carbon export slows, yet stored reserves may still move through fungal conduits toward metabolically active roots or recovering tissues. Repair processes often draw on shared nutrient pools even in darkness. The underground economy operates continuously. Philosophically, nutrient sharing reframes forest identity. Trees are not isolated producers hoarding resources for personal survival. They exist as contributors to a shared metabolic field, donors when abundant, recipients when stressed. Strength circulates. Weakness receives support. Balance emerges through exchange. Standing in a forest understory, observing thin saplings reaching patiently toward filtered light, one might assume they survive through sheer endurance alone. Yet beneath their roots, unseen currents of carbon, nitrogen, and sugar flow steadily, gifts from towering elders above. A quiet generosity encoded not in intention but in evolutionary symbiosis. The fungal networks that carry these nutrients weave cooperation into the very structure of forests. Through them, abundance softens scarcity. Through them, survival extends beyond individual capacity. And through them, the forest reveals itself not as a collection of competing trees, but as a living system sustained by shared resources, a community bound together by the slow, constant circulation of life's most essential elements. Memory, as humans understand it, is tied to the brain, to neurons firing, synapses strengthening, experiences recorded as electrical and chemical imprints that shape future behavior. It feels inseparable from consciousness, from thought, from awareness. Trees possess none of these structures. They have no brains, no neurons, no centralized processing organ where experiences gather and replay. And yet, forests behave as though they remember. Their responses to drought, fire, frost, and storm are not identical year after year. Past hardship alters future growth. Previous stress reshapes later resilience. Patterns emerge that suggest a kind of biological recollection, not conscious, but encoded in living tissue. This phenomenon is often called seasonal memory. It begins at the cellular level, in the microscopic machinery that governs plant survival. When a tree encounters environmental stress, prolonged drought, extreme cold, wildfire heat, or repeated insect attack, its cells undergo chemical and structural adjustments designed to preserve life under threat. These adjustments do not always vanish when the stress ends. Instead, they leave behind biochemical imprints that influence how the tree behaves in subsequent seasons. Drought provides one of the clearest examples. When water becomes scarce, trees activate a cascade of physiological responses. Stomata, tiny pores on leaves, close more frequently to reduce water loss. Root systems extend deeper into soil layers in search of moisture. Growth slows to conserve resources. Hormones such as abscisic acid increase, regulating stress responses across tissues. Within cells, gene expression patterns shift. Certain genes responsible for water conservation, osmotic regulation, and protective protein production become more active. Others related to rapid growth suppress their activity. If drought persists, these expression changes stabilize. Even after rainfall returns, the tree does not immediately revert to pre-drought physiology. Its stomatal sensitivity remains heightened. Its growth patterns adjust cautiously. Root expansion continues in deeper soil zones rather than shallow ones. The tree behaves as though anticipating future scarcity. This anticipation is not foresight but biochemical conditioning, the residue of stress embedded in cellular processes. Harsh winters produce similar imprints. As temperatures fall, trees undergo cold acclimation. Sugars concentrate within cells, acting as antifreeze agents that lower freezing points. Cell membranes alter lipid composition to maintain flexibility in cold conditions. Protective proteins known as dihydrins accumulate, stabilizing cellular structures against ice crystal damage. When spring arrives, these adaptations gradually reverse, yet if the previous winter was exceptionally severe, some protective pathways remain partially primed. The following autumn, cold acclimation may begin earlier or proceed more rapidly, reducing frost damage risk. The tree's tissues have, in a sense, learned. Fire exposure imprints memory differently. Low-intensity wildfires often pass through forests without killing mature trees. Heat exposure thickens bark through accelerated secondary growth. Resin production increases, improving wound sealing capacity. Bud dormancy patterns may shift, delaying leaf emergence in fire-prone seasons. Seed germination responses can also change. Some trees exposed to fire produce offspring, with higher heat tolerance or altered germination timing, adaptations shaped by parental stress exposure. These responses illustrate transgenerational memory, where environmental experiences influence not only individual trees but their descendants. At the molecular level, epigenetics plays a central role in this memory system. Epigenetic changes modify how genes express without altering the underlying DNA sequence. Chemical markers, such as methyl groups, attach to DNA strands, influencing which genes activate or remain silent. Environmental stress can trigger these epigenetic modifications. A drought may increase methylation on growth-related genes while reducing it on stress response genes. These patterns can persist long after the drought ends, shaping the tree's future physiological priorities. In some cases, epigenetic markers pass into seeds, allowing offspring to inherit stress-conditioned gene expression tendencies. Memory becomes heritable without genetic mutation. Seasonal cycles reinforce these imprints. Trees track day length, temperature shifts, and soil moisture through hormonal signaling systems. Photoperiod detection. Sensing changes in daylight duration triggers seasonal transitions such as bud formation or leaf shedding. When extreme weather disrupts these cycles, trees adjust sensitivity thresholds. After experiencing late spring frosts, for instance, some trees delay budburst in subsequent years, reducing vulnerability to repeat events. Timing becomes informed by experience. This seasonal recalibration unfolds slowly, often requiring multiple stress exposures to stabilize. A single mild drought may leave little trace. Repeated droughts over consecutive years embed stronger physiological memory. Forests located in variable climates often exhibit deeper seasonal memory than those in stable environments. Root systems store additional layers of this memory. Drought-stressed trees frequently alter root architecture, investing more energy into deeper, thicker roots capable of accessing groundwater reserves. These structural changes persist across years, influencing water uptake efficiency long after surface conditions improve. Similarly, nutrient-poor conditions may drive expanded lateral root growth, enhancing mineral acquisition zones. The soil itself retains echoes of stress. Microbial communities surrounding roots shift during drought or fire. Beneficial fungi may increase or decrease. Nitrogen-fixing bacteria populations fluctuate. Trees responding to these microbial changes adjust root exudate chemistry accordingly, shaping future soil ecosystems. Seasonal memory thus extends beyond the tree into its environmental partnerships. Electrical signaling contributes subtle reinforcement. Stress events alter membrane voltage patterns in plant cells. These changes influence ion transport, hormonal movement, and gene activation pathways. Repeated stress exposures refine electrical responsiveness, allowing faster signaling during subsequent threats. Though far slower than animal neural responses, these electrical adjustments add another dimension to plant memory encoding. Resource allocation strategies also shift through experience. After surviving severe stress, trees often prioritize storage over rapid growth. Carbon reserves accumulate in roots and trunks rather than fueling canopy expansion. This conservative strategy improves survival odds if hardship returns. In contrast, trees raised in stable environments invest more aggressively in growth, sometimes at the expense of resilience. Experience shapes risk tolerance. Seasonal memory also influences reproductive timing. Trees exposed to prolonged drought may reduce seed production temporarily, conserving energy. When favorable conditions return, reproductive output may surge, a delayed but amplified response linked to stored reserves accumulated during stress recovery. Flowering and pollination cycles adjust accordingly. Insects and wildlife interacting with these trees indirectly experience the ripple effects of seasonal memory, as nectar availability, fruiting patterns, and habitat structure shift in response. At the forest scale, these individual memories accumulate into ecosystem behavior. After widespread drought, entire forests may display synchronized growth suppression the following year. After major fires, regeneration patterns align with stress-conditioned seed responses. Winter severity influences collective budburst timing across species clusters. The forest behaves like a distributed memory system, experience encoded across millions of trees simultaneously. Human observation of these patterns dates back centuries, though scientific understanding arrived much later. Foresters noted that trees grown from seeds collected in harsh climates often displayed superior resilience when planted elsewhere. This led to early recognition that environmental experience influenced plant behavior beyond genetics alone. Modern molecular biology later revealed the epigenetic mechanisms underlying these observations. Climate change now places unprecedented importance on seasonal memory. As weather patterns grow more erratic, droughts intensifying, winters fluctuating, fires increasing, trees rely heavily on stress-conditioned responses. Forests with deeper seasonal memory adapt more readily to rapid change than those historically buffered by stable climates. Yet adaptation has limits. If environmental shifts outpace the speed at which trees can encode and transmit memory, resilience may falter. Saplings lacking parental stress exposure may struggle in newly harsh conditions. Thus, preserving older trees, repositories of long-term environmental memory, becomes ecologically vital. They carry encoded histories of past droughts, past fires, past winters, biological archives embedded in living tissue. Even fungal networks participate in seasonal memory. Mycorrhizal communities adjust composition based on environmental stress. Drought-resistant fungal species may proliferate after prolonged dry periods, enhancing water acquisition capacity for connected trees in subsequent seasons. This fungal adaptation complements plant memory, reinforcing ecosystem resilience. Night and dormancy phases also contribute to memory consolidation. During winter dormancy, trees reorganize cellular structures, repair damage, and stabilize epigenetic markers established during the growing season. Memory becomes chemically fixed during these resting periods. Spring reactivation then unfolds along pathways shaped by past experience. Philosophically, seasonal memory blurs distinctions between awareness and adaptation. Trees do not recall events consciously, yet their tissues carry histories. Their growth rings record drought years in narrow bands. Fire scars etch thermal trauma into wood. Cellular chemistry reflects survival battles invisible to human eyes. Memory exists without mind. Standing before an ancient tree, one stands before centuries of recorded experience, encoded in bark thickness, root depth, gene expression, and epigenetic markers layered across time. Each harsh winter survived refines cold tolerance. Each drought endured deepens water strategy. Each fire weathered reshapes bark and bud behavior. The tree becomes its history, and when seeds fall from its branches, fragments of that history travel forward, subtle biochemical inheritances guiding new growth in landscapes shaped by old hardship. Thus, forests do not face each season as blank slates. They enter spring informed by winter. They enter drought informed by rain. They enter fire informed by flame. A slow accumulation of environmental memory guiding survival, silent, cellular, enduring, written not in thought, but in the living chemistry of wood, leaf, and root. Beneath the forest floor, where light dissolves into darkness and roots replace branches, another form of communication moves quietly through living tissue, one that does not rely on chemicals alone. It is electrical. For much of human history, electricity was considered the domain of animals, a force associated with nerves, muscles, and brains. The discovery that electrical impulses traveled along neurons, transmitting sensation and thought, reinforced the idea that such signaling required specialized organs and conscious awareness. Trees possess neither. They have no brains, no centralized nervous systems, no neurons firing in rapid succession. Yet research over the past century has revealed that plant tissues, including roots, generate and transmit electrical signals that bear striking conceptual similarities to neural impulses, slower, simpler, but functionally meaningful. To understand this phenomenon, one must first look at the cellular structure of plants. Every plant cell maintains an electrical gradient across its membrane. Ions, charged particles such as potassium, calcium, and chloride, are unevenly distributed between the interior of the cell and the surrounding environment. This imbalance creates a voltage difference known as membrane potential. In stable conditions, this voltage remains relatively constant. But when a plant experiences stress, mechanical damage, temperature shock, drought, or chemical disturbance, ion channels in cell membranes open or close rapidly. Charged particles rush across the membrane, altering voltage patterns. This change can propagate from cell to cell, creating a traveling electrical wave. In animals, such waves move along specialized neurons at remarkable speeds. In plants, the movement is slower, passing through vascular tissues, plasmodesmata, and root junctions. Still, the principle remains: a disturbance in one location can generate an electrical signal that spreads through the organism. Roots, embedded in complex soil environments, are particularly active in this signaling. They constantly encounter variations in moisture, mineral concentration, microbial presence, and physical barriers. Electrical signals help coordinate responses, directing growth toward nutrient-rich zones or away from toxins. When a root tip detects drought conditions, for example, electrical changes initiate hormonal cascades that travel upward through the plant. Leaves may close stomata, growth rates adjust, resource allocation shifts towards survival rather than expansion. Electrical communication thus links underground sensing to above-ground response. In forest ecosystems, these signals extend beyond individual trees. Root systems often graft together when growing in close proximity. These grafts fuse vascular tissues, creating direct physiological bridges. Through these connections, electrical impulses generated in one tree can pass into another. Fungal networks further amplify this transmission potential. Mycorrhizal fungi, already known for nutrient and chemical signal exchange, also conduct electrical fluctuations. Hyphal filaments maintain ionic gradients similar to plant cells. When electrical signals enter fungal tissues through root interfaces, they may propagate along fungal threads toward connected trees. The speed of transmission remains slow, measured in millimeters per second rather than meters, but forests operate on timescales where such pacing remains effective. An insect attack on one tree may trigger electrical disturbances that travel through root grafts and fungal pathways, priming defensive responses in neighbors before chemical signals fully diffuse. Electrical signaling often precedes chemical signaling in timing, functioning as an early alert layer. Mechanical injury produces some of the clearest electrical responses. When a leaf is torn or bark punctured, electrical potentials shift instantly. This signal moves along vascular tissues, activating defensive gene expression far from the wound site. Studies have recorded voltage changes traveling from damaged leaves to undamaged roots within minutes. Roots receiving this signal alter ion transport, hormone production, and metabolic priorities, coordinating systemic response. Temperature extremes generate similar impulses. Rapid cooling or heating alters membrane fluidity, triggering ion channel activity. Electrical waves spread through roots, initiating cold acclimation or heat stress responses before tissue damage becomes severe. This capacity allows trees to adjust physiology preemptively as environmental conditions shift. Drought detection relies heavily on root-based electrical signaling. As soil moisture declines, root cells experience osmotic stress. Ion flux changes trigger voltage variations that propagate upward, activating abscisic acid production, the hormone responsible for stomatal closure. Leaves receiving this hormonal message reduce water loss, preserving internal hydration. Without electrical signaling linking root detection to leaf response, such coordination would lag dangerously behind environmental change. Electrical pulses also influence growth direction. Root tips navigate soil through tropisms, directional growth responses to stimuli such as gravity, moisture, and nutrients. Electrical gradients within root tissues help regulate cell elongation patterns, guiding roots toward favorable conditions. When electrical disturbances signal toxic substances or physical barriers, growth pathways adjust accordingly. The root system becomes both sensor and responder. In interconnected forests, these impulses accumulate into distributed signaling networks. Multiple trees experiencing stress simultaneously may generate overlapping electrical waves within shared fungal and root infrastructures. The network's electrical activity reflects collective environmental sensing, a slow, pulsating field of bioelectrical communication. Though far removed from neural cognition, the resemblance to nervous systems remains conceptually striking. Animals use electrical signals to transmit sensation and coordinate response. Trees use electrical signals to detect stress and regulate physiology. The difference lies in speed and complexity, not in foundational mechanism. Scientific exploration of plant electrophysiology dates back to the 19th century. Researchers such as Jagadish Chandra Bose conducted early experiments measuring electrical responses in plants using sensitive instruments. His work demonstrated that plants exhibited measurable electrical reactions to stimuli, mechanical, thermal, and chemical. Though initially met with skepticism, later research confirmed and expanded his findings. Modern microelectrode technology allows scientists to record electrical activity within roots and stems with precision. Voltage fluctuations corresponding to stress events, nutrient uptake, and growth processes have been mapped extensively. The forest floor, once thought electrically inert, reveals itself as alive with slow-moving bioelectrical currents. Electrical signaling interacts closely with hormonal pathways. Voltage changes often trigger the release or transport of plant hormones, jasmonic acid for defense, auxins for growth regulation, abscisic acid for drought response. Electrical waves thus act as initiators, while chemical messengers execute prolonged physiological adjustments. Together they form layered communication systems, fast electrical alerts followed by slower chemical responses. Night cycles influence these dynamics subtly. Electrical activity in roots does not cease in darkness. While photosynthetic signals diminish, root sensing continues. Moisture shifts, microbial interactions, and temperature changes still generate voltage fluctuations guiding nocturnal metabolic adjustments. The underground electrical network never fully rests. Environmental pollutants can disrupt these signals. Heavy metals, soil salinity, and chemical contaminants alter ion gradients, interfering with electrical transmission. Trees exposed to such disturbances may exhibit impaired signaling, slower stress responses, reduced growth coordination, and weakened resilience. Protecting soil health thus preserves not only nutrient exchange but electrical communication integrity. Even fungal partners contribute actively to bioelectrical processes. Hyphae exhibit electrical oscillations linked to nutrient transport and growth patterns. Some studies suggest fungal networks display synchronized electrical rhythms, potentially coordinating metabolic activity across large areas. When connected to trees, these rhythms may integrate into broader forest signaling systems. The wood wide web, therefore, carries not only chemistry but electricity, a dual-channel communication infrastructure. Philosophically, this blurs distinctions between plant and animal signaling paradigms. While trees lack consciousness, their electrical responsiveness suggests a form of environmental awareness embedded in cellular function. They detect injury, stress, and resource gradients through voltage changes, responding in coordinated, adaptive ways. The forest becomes electrically perceptive, sensing through roots what animals sense through nerves. Lightning strikes illustrate electrical vulnerability and adaptation. Trees struck by lightning experience massive electrical surges, often fatal. Yet surrounding trees detect resulting soil ionization changes through root systems, adjusting growth and signaling patterns in response to altered mineral availability. Even catastrophic electrical events ripple into ecological communication. Seasonal changes modulate baseline electrical activity. Spring growth increases ion transport as nutrient uptake accelerates. Autumn dormancy reduces electrical fluctuations as metabolic processes slow. Winter freezes dampen conductivity, but do not eliminate residual activity in unfrozen tissues. Electrical signaling follows the rhythm of life cycles. Standing in a forest, one perceives stillness, trunks unmoving, leaves rustling softly. Yet underground, slow electrical pulses travel root to root, fungus to tree to tree, invisible currents coordinating survival across living networks. Not thoughts, not emotions, but signals encoded in ion flux, membrane voltage, and conductive tissues shaped by evolution. They move patiently. They move silently. And though they lack the speed of neurons or the awareness of minds, they carry meaning, linking the hidden lives of trees in a quiet electrical dialogue, unfolding continuously beneath the soil. Beneath the forest floor, where roots stretch into darkness and soil, holds the layered residue of centuries. Fungi move quietly through spaces too small for roots to enter and too complex for humans to easily imagine. Their filaments, thin as threads of smoke, weave through grains of earth, wrapping stones, penetrating decaying wood, and encasing the tips of living roots. To see them under magnification is to witness an intricate lacework of life, branching, merging, dissolving, and reforming in response to the needs of the forest. For a long time, these fungal presences were understood primarily as connectors, biological cables linking trees into shared networks. But deeper research has revealed that their role extends far beyond passive connection. Fungi do not merely allow exchanges to occur. They shape them, regulate them, prioritize them. In the underground economy of the forest, fungi act as intermediaries, brokers, translators, and governors of resource flow. Their involvement begins at the root interface. When fungal hyphae encounter a tree root, they form symbiotic structures known as mycorrhiza. In some cases, They sheath the root externally, forming a dense mantle. In others, they penetrate root cells, creating branching exchange structures within. These interfaces function as biochemical marketplaces, sites where nutrients and carbon are traded. The tree supplies sugars, glucose, sucrose, and other carbohydrates produced through photosynthesis. The fungus, unable to photosynthesize, depends on these carbon compounds as its primary energy source. In return, the fungus supplies minerals absorbed from soil: phosphorus, nitrogen, potassium, and trace elements. Yet the exchange is not equal by default. Fungi regulate how much nutrient they provide based on how much carbon they receive. If a tree offers abundant sugars, fungal transport channels widen, mineral flow increases, and the partnership strengthens. If carbon supply declines, fungal nutrient transfer diminishes accordingly. This reciprocal negotiation positions fungi as economic mediators rather than passive conduits. When multiple trees connect to the same fungal network, the intermediary role becomes more complex. Carbon entering the network from one tree does not automatically flow equally to all others. Fungi allocate resources selectively, guided by biochemical signals, environmental context, and evolutionary incentives. A healthy tree supplying abundant carbon may indirectly subsidize weaker trees through fungal redistribution. But fungi often retain a portion of transferred carbon for their own growth and metabolic maintenance. They are participants in the exchange, not neutral pipelines. Nutrient distribution likewise reflects fungal decision-making. If a tree rooted in nutrient-poor soil contributes high carbon, fungi may prioritize delivering phosphorus or nitrogen to that tree, reinforcing the partnership. Trees contributing little carbon may receive reduced nutrient support, a regulatory mechanism preventing exploitation. This balancing function stabilizes the network. Fungi also interpret chemical signals moving through the system. When a tree releases distress compounds due to insect attack or drought stress, these molecules enter fungal tissues. The fungus detects them through receptor systems sensitive to hormonal and metabolic changes. Rather than simply passing the signal unchanged, fungi may amplify, dampen, or redirect it. For instance, if distress signals indicate localized stress affecting only one tree, fungi may route warnings primarily to nearby connected trees most at risk. If the stress reflects broader environmental change, such as drought, signal distribution may spread more widely across the network. In this way, fungi act as translators, interpreting chemical languages produced by trees and deciding how extensively to broadcast them. Electrical signaling introduces further intermediary roles. Fungal hyphae conduct ionic currents associated with electrical pulses traveling through roots. When these signals enter fungal tissues, conduction patterns may alter depending on hyphal density, moisture levels, and fungal metabolic state. Electrical impulses may accelerate or decelerate depending on fungal pathway characteristics, influencing how quickly warnings or stress responses propagate across the forest web. Fungi thus shape both chemical and electrical communication flow. Their regulatory influence extends into defense coordination. When one tree activates toxin production in response to herbivory, neighboring trees connected through fungal networks may begin similar responses. Fungi help distribute the initiating signals, ensuring that defensive chemistry spreads efficiently across connected hosts. Yet fungi may also moderate excessive responses. Producing defensive toxins requires energy that ultimately flows into fungal partners. If defensive activation becomes widespread without sufficient carbon supply, fungal metabolism may suffer. Balancing defense signaling with metabolic sustainability becomes part of fungal mediation. Fungi also influence which trees connect most strongly within the network. Different fungal species prefer specific host trees. Some form exclusive partnerships, others maintain broad host ranges. These preferences create sub-networks within the larger web, clusters of trees more tightly linked through shared fungal symbionts. Resource and signal flow intensifies within these clusters. Thus, fungal species diversity shapes communication architecture across forests. Older fungal colonies often form the most stable intermediary hubs. Having persisted for decades or centuries, their hyphal networks span vast soil regions. They connect mature trees, saplings, shrubs, and decomposing matter simultaneously. Their regulatory reach extends across multiple ecological niches. These ancient fungal systems function as infrastructural backbones of forest exchange. Decomposition deepens fungal intermediary roles. When trees die, fungi colonize their decaying roots and wood. Nutrients released through decomposition enter fungal tissues, then redistribute into living plant partners. In this context, fungi translate death into nourishment, converting structural carbon and minerals from fallen trees into usable resources for survivors. They mediate life cycles as much as they mediate communication. Environmental stress reshapes fungal brokerage behavior. During drought, fungal hyphae shrink or redirect growth toward moist soil pockets. Resource transport pathways reorganize accordingly. Trees connected through these fungi receive water and nutrients along reconfigured routes, reflecting environmental constraints. Fungal adaptability ensures network resilience. In nutrient-rich conditions, fungi may expand connections, linking additional trees and increasing exchange complexity. In scarcity, they may retract, prioritizing partnerships offering higher carbon return. This dynamic restructuring resembles market regulation responding to supply and demand. Some fungi even manipulate plant behavior to enhance their own survival. Research suggests certain Mycorrhizal species influence host root growth patterns, encouraging expansion into zones favorable for fungal proliferation. Others may alter host carbon allocation, directing more sugars toward root systems feeding fungal partners. These manipulations blur boundaries between cooperation and subtle exploitation. Still, the net outcome often benefits both participants, sustaining the broader ecosystem. Fungi also filter pathogens within the network. When harmful microbes attempt to spread through root interfaces, fungal mantles can block entry. Antimicrobial compounds produced by fungi protect host trees from soil-borne diseases. This protective mediation adds another layer to their intermediary function. They regulate not just exchange but security. Communication fidelity depends heavily on fungal health. Soil pollution, acidification, and climate stress can damage fungal communities. When intermediary systems weaken, nutrient sharing declines, warning signals slow, and forest resilience diminishes. Restoring fungal diversity becomes essential to restoring network functionality. Modern forestry increasingly recognizes this. Reforestation projects now introduce mycorrhizal inoculants alongside tree planting. Without fungal intermediaries, young trees struggle to establish robust exchange networks, leading to slower growth and higher mortality. Technology begins to partner intentionally with fungal infrastructure long taken for granted. Fungi's intermediary role extends into seasonal cycles. During spring growth, carbon flow from trees into fungal networks surges, fueling hyphal expansion. Nutrient transport accelerates, supporting leaf emergence and shoot elongation. In autumn, carbon input declines. Fungi shift towards storage and decomposition, redistributing nutrients from falling leaves back into root systems. They regulate seasonal nutrient recycling rhythms. Even electrical rhythms pass through fungal mediation. Recent research has observed oscillatory electrical patterns within fungal networks, fluctuations potentially linked to nutrient pulses and environmental sensing. These oscillations may synchronize resource flow across connected trees, adding temporal coordination to exchange systems. The forest web pulses not only chemically but electrically, guided in part by fungal activity. Philosophically, fungi challenge anthropocentric models of intelligence and communication. They lack brains yet coordinate complex exchanges. They lack intention, yet regulate systemic balance. Their decisions emerge from biochemical feedback loops shaped by evolutionary pressures rather than conscious strategy. Still, their function resembles governance. They allocate resources, interpret signals, balance partnerships, and sustain network stability across vast ecological systems. Without them, the wood wide web would collapse into disconnected individuals, trees isolated from one another, deprived of shared nutrients and collective warning systems. Fungi are not background participants. They are the translators of forest language, the brokers of subterranean economies, the interpreters of chemical distress, and the regulators of exchange. They decide how much flows, where it flows, and when. In darkness beyond sight, their hyphae carry whispers of carbon, nitrogen, hormones, and ions, messages passing through living threads that shape survival above ground. And so, while trees stand tall in sunlight, appearing as the visible actors of the forest, it is often the unseen fungi below that orchestrate the deeper conversation, moderating the flow of life itself through patient, silent mediation. Forests rarely grow as single-species collectives. To walk through a natural woodland is to move through diversity layered in height, texture, and biological strategy. Birch trees with pale bark stand beside dark conifers. Pines lift narrow crowns above spreading maples. Firs grow in cool shadows beneath deciduous canopies. Each species carries its own evolutionary history, its own tolerances, its own rhythms of growth and dormancy. For centuries, this diversity was interpreted primarily through competition, different species vying for light, water, and soil nutrients. Mixed forests were thought to represent ecological balance, only because no single species had yet outcompeted the others. But beneath the soil, where roots interlace and fungal networks weave invisible corridors, diversity expresses itself not just through rivalry, but through alliance. Different tree species communicate and share resources through shared mycorrhizal networks, forming what might be described as interspecies partnerships, collaborative systems that enhance survival across ecological gradients. These connections begin with fungi. Mycorrhizal fungi do not restrict themselves to single hosts. Many species form associations with multiple tree types simultaneously. A single fungal network may connect birch, fir, pine, spruce, and understory shrubs all at once, linking deciduous and coniferous lineages through shared hyphal pathways. Within these pathways, nutrients and chemical signals move without strict regard for species boundaries. Carbon exchange offers one of the clearest demonstrations. Birch trees, deciduous and fast-growing, leaf out early in spring. Their broad leaves capture sunlight efficiently during the first warm weeks of the year, producing abundant photosynthate sugars generated through photosynthesis. Conifers such as fir and pine, by contrast, retain needles year-round but often photosynthesize more slowly in early spring due to temperature limitations. Through fungal networks, birch trees may transfer carbon to neighboring conifers during this seasonal window, supplementing their metabolic needs while their own photosynthetic output remains limited. As seasons shift, the dynamic reverses. In autumn, when birch leaves senesce and fall, photosynthesis ceases. Evergreen conifers, still bearing needles, continue producing sugars during mild conditions. Carbon may then flow from fir or pine back into birch root systems or nearby deciduous saplings preparing for winter dormancy. Reciprocity unfolds through seasonal complementarity. This exchange stabilizes mixed forests, ensuring that energy deficits in one species during certain periods are buffered by surplus production in another. Nutrient sharing follows similar pathways. Different tree species access soil resources differently. Some extend deep taproots into groundwater zones. Others spread shallow roots across nutrient-rich topsoil. Fungal intermediaries absorb minerals from both zones, redistributing nitrogen, phosphorus, and micronutrients among connected trees regardless of species. A pine rooted in sandy, nitrogen-poor soil may receive nitrogen compounds via fungi linked to nearby birch accessing richer organic layers. In return, pine-derived carbon may sustain fungal growth that benefits birch nutrient acquisition later. The network balances resource disparities. Signal communication transcends species lines as well. When insects attack one species, distress signals released into fungal pathways can alert entirely different species connected to the same network. A beetle infestation on spruce may trigger defensive chemical activation in nearby fir or pine, even if those species are not the primary target. This cross-species warning increases forest resilience. Volatile airborne compounds reinforce this system. Trees release species-specific blends of volatile organic compounds when stressed, yet many of these molecules are detectable across species boundaries. A deciduous tree sensing conifer distress volatiles may still activate generalized defense pathways, strengthening cell walls, producing deterrent compounds, or adjusting growth priorities. Communication operates through shared biochemical languages rather than isolated dialects. Fungal mediation ensures that signals maintain coherence across species differences. Because fungi interact biochemically with multiple hosts, they function as translators, converting host-specific chemical cues into network-compatible signals. This allows trees with distinct evolutionary chemistries to participate in shared warning systems. The forest becomes an integrated sensory network. Water redistribution also occurs interspecifically. During drought, deep-rooted species may access subsurface moisture unavailable to shallow-rooted neighbors. Through fungal conduits, water gradients facilitate transfer, alleviating dehydration stress in species less equipped for deep extraction. This hydraulic cooperation reduces mortality during prolonged dry periods. Root grafting between species adds another layer of connectivity, though less common than fungal mediation. Physical root fusions between compatible species allow direct nutrient and signal exchange, reinforcing interspecies alliances where they occur. Forest structure reflects these hidden partnerships. Mixed forests often display higher resilience to disease and environmental stress than monocultures. Cross-species communication distributes warning signals more widely. Nutrient redistribution buffers localized soil deficiencies. Pest outbreaks spread more slowly when host diversity complicates insect specialization. Diversity strengthens network intelligence. Fungal network topology adapts to species composition. Certain fungi specialize in connecting deciduous hosts, others coniferous. In mixed forests, overlapping fungal communities create layered networks, some channels favoring birch to birch exchange, others bridging birch to pine, pine to fir, and so on. The result is a multi-dimensional communication grid spanning taxonomic boundaries. Seasonal memory integrates into this interspecies system. If one species develops stress-conditioned resilience, drought tolerance for example, its altered chemical signaling may prime neighboring species through fungal transmission. Defensive readiness spreads not just genetically, but ecologically. Experience diffuses through connection. Seedling establishment benefits from cross-species networks as well. Young trees germinating beneath mixed canopies often connect first to fungal partners, already linked to mature trees of other species. Through these connections, they receive carbon supplementation, nutrient access, and environmental signaling advantages before forming species-specific partnerships. The network welcomes new participants regardless of lineage. Decomposition further reinforces interspecies exchange. When trees die, fungi break down their tissues, redistributing nutrients across the network. A fallen birch may feed pine seedlings. A decaying fir may nourish maple saplings. Death dissolves species boundaries, converting individual biomass into shared ecosystem resource. The forest recycles itself cooperatively. Human forestry practices have illuminated the importance of these alliances. Monoculture plantations, forests composed of a single species, often display reduced resilience. Pest outbreaks spread rapidly. Soil nutrients deplete unevenly. Fungal diversity declines, weakening network connectivity. In contrast, mixed species forests exhibit stronger nutrient cycling, better drought tolerance, and improved regeneration rates. Cross-species communication underpins this resilience. Climate change magnifies its importance. As environmental stress intensifies, forests relying on single-species strategies struggle to adapt. Mixed forests, with their interspecies nutrient sharing and signaling networks, buffer ecological shocks more effectively. Energy surplus in one species can offset deficit in another. Stress signals spread more widely, enhancing collective preparedness. Even wildlife interactions reflect these alliances. Animal species dependent on diverse forest structures benefit indirectly from tree cooperation. Nutrient sharing supports understory growth, creating habitats for insects, birds, and mammals. Signal-mediated defense reduces catastrophic defoliation events that could destabilize entire ecosystems. Tree alliances ripple outward through trophic layers. Fungal intermediaries remain central to maintaining these relationships. By connecting species chemically and electrically, they enable communication that would otherwise be impossible. Their selective mediation ensures that exchanges remain balanced rather than exploitative. They are the architects of interspecies dialogue. Philosophically, cross-species communication challenges anthropocentric assumptions about individuality and competition in nature. Forests reveal that survival often depends on cooperation across genetic boundaries. Trees do not confine assistance to their own species. They participate in broader ecological partnerships shaped by shared environment and mutual benefit. Strength lies in connection. Standing within a mixed forest, one might see diversity in bark color, leaf shape, and canopy structure. Yet beneath the soil, those differences blur into a shared metabolic and communicative field. Carbon flows from birch to fir, nitrogen moves from pine to spruce, warnings travel from maple to cedar, signals, nutrients, and water circulate through fungal threads that ignore species borders entirely. The forest functions not as separate populations, but as an integrated alliance, a living network where diversity enhances resilience and communication bridges evolutionary distance. A quiet coalition rooted in darkness, sustained by exchange, and bound together by the slow, patient language of interconnected life. Connection in a forest is rarely free. Beneath the soil, where fungal networks weave living corridors between roots, resources move in quiet circulation: carbon, nitrogen, sugars, water, defensive signals. These exchanges sustain saplings in shade, stabilize drought-stressed neighbors, and reinforce the resilience of entire ecosystems. From a distance, the system appears cooperative, almost generous, as though trees support one another in a communal effort toward survival. Yet every transfer carries a cost. When a healthy, sunlit tree channels carbon into fungal networks, that carbon does not remain available for its own immediate growth. Sugars produced through photosynthesis represent stored sunlight, energy that could thicken bark, extend roots, produce seeds, or expand canopy reach. Redirecting a portion of that energy outward reduces what remains for the donor's individual advancement. The loss is rarely catastrophic, but it is measurable. Growth rings in trees subjected to heavy resource redistribution sometimes narrow slightly, reflecting slower trunk expansion. Seed production may decline during years of high carbon transfer. Root development may prioritize network maintenance over territorial expansion. Connection, then, involves trade-offs. Fungi themselves require compensation. Acting as intermediaries, they consume carbon supplied by host trees to fuel their own metabolism, growth, and nutrient transport activity. The fungal web is not a free infrastructure. It is sustained through continuous energy input. Thus, when a donor tree shares resources, some portion supports recipient trees, while another portion sustains the fungal brokers facilitating exchange. The donor invests in the system as much as in its neighbors. This dynamic raises a profound ecological question: why would a tree, shaped by evolutionary pressures favoring survival and reproduction, allocate resources in ways that appear to weaken itself? The answer lies not in altruism as humans define it, but in layered survival strategy. Forests function as interdependent systems. A tree's long-term survival depends heavily on the stability of its surrounding ecosystem, soil health, fungal diversity, microclimate regulation, and structural protection from wind or erosion. Supporting neighboring trees helps maintain these conditions. When mature trees transfer carbon to shaded saplings, they ensure future canopy continuity. A dense forest moderates temperature extremes, retains soil moisture, and shields individuals from storm damage. If surrounding vegetation collapses, Isolated trees face harsher environmental exposure. Resource sharing thus reinforces environmental buffering. Kin selection further influences transfer patterns. Trees often preferentially allocate more carbon to genetically related saplings connected through fungal networks. Chemical signaling at root interfaces allows partial recognition of kinship, shaping resource distribution gradients. Supporting offspring increases the donor's genetic legacy, ensuring that its lineage persists even after death. In this context, resource sharing becomes evolutionary investment rather than selfless sacrifice. Still, transfers extend beyond kin. Cross-species exchanges occur regularly, suggesting that broader ecosystem stability also drives sharing behavior. Supporting non-related neighbors maintains fungal networks, soil integrity, and nutrient cycling systems upon which the donor tree itself depends. Cooperation enhances collective resilience, which feeds back into individual survival probability. Environmental stress amplifies the cost-benefit calculation. During drought, a donor tree may transfer water or carbon to stressed neighbors through fungal conduits. This weakens its own reserves, but reduces the likelihood of widespread mortality that could destabilize soil and microclimate conditions. If neighboring trees perish, increased sunlight exposure may raise soil temperatures, accelerating moisture loss and harming the donor in subsequent seasons. Sharing mitigates systemic collapse. Fungal mediation ensures that donors are not endlessly exploited. Fungi regulate exchange through reciprocal gating mechanisms. Trees contributing high carbon receive enhanced nutrient access. Those contributing little may experience reduced fungal connectivity or mineral transfer. This reciprocity maintains equilibrium within the network. Still, imbalance can occur. Parasitic plants and low-carbon contributors may draw resources disproportionately. Fungal partners often respond by limiting connections, but during early infestation stages, donor trees may incur noticeable resource drain. The cost of connection becomes temporarily asymmetrical. Energy diverted toward defense signaling adds further burden. When one tree experiences insect attack and transmits warning signals through fungal networks, neighboring trees activate toxin production preemptively. Producing these compounds requires carbon and nitrogen investment, resources that could otherwise fuel growth. If threats do not materialize, the energy spent on defense becomes a lost opportunity. Yet failing to respond risks catastrophic damage if herbivores spread. Connection forces trees to balance preparedness against efficiency. Seasonal cycles influence this calculus. During peak photosynthesis in summer, carbon surpluses make sharing less burdensome. During early spring or late autumn, when energy reserves run lower, transfers may tighten as donor trees prioritize internal needs. Fungal intermediaries help regulate these seasonal adjustments, reducing excessive drain during scarcity. Mother trees often bear the highest connection costs. As central hubs in fungal networks, they maintain the most extensive partnerships and resource flows. Their large canopies generate significant carbon, but their redistribution responsibilities also scale accordingly. Despite this, mother trees tend to survive longer than many isolated counterparts, suggesting that the systemic stability they help maintain offsets individual cost burdens. Connection sustains longevity through ecosystem buffering. Storm resilience illustrates this principle. Trees embedded within dense, interconnected forests experience less wind stress than solitary individuals. Neighboring trunks break gust force, root systems interlock soil, and fungal networks enhance nutrient stability. A tree that shared resources contributing to forest density benefits structurally during extreme weather. Investment returns through environmental protection. Fire ecology presents similar dynamics. Healthy forests with intact networks recover faster after low-intensity fires. Resource sharing supports regeneration, preventing erosion and maintaining fungal viability. Donor trees that survive the fire benefit from quicker ecosystem stabilization. Connection accelerates recovery. Evolutionary design underpins these patterns. Natural selection operates not only at the level of individual organisms, but within ecological networks. traits promoting cooperative resource exchange persist when they enhance overall survival probability across generations. Trees capable of forming extensive fungal partnerships and sharing resources effectively may leave more descendants over long timescales than competitive individuals that destabilize their surroundings. Cooperation becomes adaptive. Yet the question of altruism remains philosophically compelling. Trees do not choose to share resources in conscious generosity. Their exchanges arise from biochemical feedback loops, symbiotic dependencies, and evolutionary conditioning rather than moral intention. Still, the functional outcome resembles generosity, energy flowing from abundance to scarcity, strength supporting weakness. It is altruism without awareness. The cost also manifests structurally. Maintaining fungal partnerships requires continuous carbon supply. Fungal mantles around roots demand metabolic upkeep. Hyphal proliferation depends on steady energy input. Trees deeply embedded in networks allocate more carbon below ground than isolated trees, a hidden expenditure invisible above the soil surface. Yet this underground investment enhances nutrient acquisition efficiency, offsetting costs through improved mineral access. Even electrical signaling carries metabolic expense. Generating and propagating electrical impulses requires ion transport across membranes, consuming cellular energy. Coordinating stress responses through electrical channels diverts resources from growth functions. Connection always exacts some physiological price. Human interference often magnifies these costs. When forests fragment due to logging or land development, remaining trees retain fungal obligations but lose reciprocal support from removed neighbors. Resource sharing becomes one-sided, straining donor trees already coping with environmental disruption. Network imbalance accelerates decline. Conversely, intact forests distribute cost more evenly across participants, maintaining sustainable exchange patterns. The system relies on density and diversity to remain energetically viable. Time perspective reframes the cost entirely. A tree may live centuries. Resource sharing that slightly slows annual growth but stabilizes ecosystem function over centuries ultimately enhances lifetime survival and reproductive success. Short-term loss becomes long-term gain. Even death transforms cost into contribution. When a donor tree falls, its stored carbon and nutrients enter fungal redistribution channels, nourishing surrounding vegetation. The lifetime energy investment it made in the network continues circulating after its physical form decays. Connection transcends lifespan. Philosophically, the cost of connection dissolves rigid boundaries between self and system. A tree does not survive purely as an isolated organism. Its health depends on fungal partners, neighboring vegetation, soil microbes, and environmental stability, all sustained partly through shared resource exchange. Weakening slightly to strengthen the whole becomes evolutionarily rational. Standing within a forest, one sees towering trunks reaching skyward, each appearing self-sufficient. Yet beneath the soil energy flows outward from those giants, sugars, nitrogen, water, supporting lives unseen. Their strength is not diminished without purpose. It circulates, and through that circulation, forests endure, not as collections of competing individuals, but as interconnected systems where the cost of connection becomes the foundation of collective survival. For most of human history, forests were understood through what could be seen. Trunks, branches, leaves, roots exposed by erosion. These were the visible components shaping scientific interpretation. Early botanists cataloged species, measured growth rings, studied leaf structures, and mapped canopy competition. Trees were classified as individuals sharing space but not substance, their interactions limited to shading, root overlap, and competition for water and minerals. The soil beneath them remained largely mysterious. It was known to contain roots and microbes, but the idea that forests possessed integrated communication systems, exchanging nutrients and signals across species boundaries, lay far beyond the conceptual frameworks of early science. The confirmation of underground forest networks required technological advances capable of revealing processes invisible to human senses. One of the most transformative tools in this discovery was isotopic tracing. Isotopes are atoms of the same element that differ slightly in mass due to extra neutrons. Some isotopes occur naturally, others can be introduced experimentally. Because isotopes can be detected with high precision, they serve as markers, allowing scientists to track the movement of specific elements through biological systems. In forest research, scientists began by introducing carbon isotopes into individual trees. This was often done by enclosing a branch or canopy section in a sealed chamber and exposing it to carbon dioxide enriched with a rare isotope, such as carbon-13 or radioactive carbon-14. Through photosynthesis, the tree incorporated this labeled carbon into sugars, distributing it internally through phloem tissues. The expectation under older models of tree individuality was that the isotope would remain confined within the treated tree. But when researchers later sampled neighboring trees connected underground, they detected the labeled carbon within them as well. The isotope had moved, transferred through unseen pathways linking root systems. Further experiments refined this finding. By labeling one species such as birch, and later detecting the isotope in different species like fir or pine, scientists confirmed that carbon exchange crossed species boundaries. These transfers occurred even when physical root contact was prevented by mesh barriers that allowed fungal filaments through but blocked roots. The conclusion became unavoidable. Fungal networks mediated the exchange. Isotopic tracing expanded beyond carbon. Nitrogen isotopes were introduced into soil near specific trees. Later detection of labeled nitrogen in distant, connected trees demonstrated mineral redistribution through shared networks. Phosphorus tracing revealed similar patterns. Each experiment reinforced the reality of underground resource sharing. While isotopes revealed movement, they did not show structure. To map the physical architecture of these networks, scientists turned to soil analysis and microscopy. Soil samples collected from forest floors were examined under high magnification imaging systems. What emerged was astonishing. Dense webs of fungal hyphae threading through soil particles, wrapping roots, branching into complex three-dimensional lattices. Staining techniques allowed researchers to differentiate fungal species and visualize connection points where hyphae fused with root cells. These microscopic observations transformed understanding of forest structure. Roots were no longer isolated strands probing soil independently. They were embedded within fungal mantles, integrated interfaces where plant and fungus merged functionally. Advanced imaging, including scanning electron microscopy, revealed exchange structures such as arbuscules, tiny tree-shaped fungal formations within root cells where nutrient transfer occurred. The forest communication infrastructure began to take visible form. Micro-sensors introduced yet another layer of discovery. Sensitive electrodes inserted into plant tissues allowed scientists to detect electrical fluctuations associated with stress signaling. By placing microelectrodes in roots, stems, and fungal substrates simultaneously, researchers observed electrical impulses propagating through interconnected systems. These impulses traveled from injured plants to neighbors, confirming that communication extended beyond chemical diffusion. Soil-embedded sensors measured nutrient gradients, moisture flux, and ion movement across fungal pathways. Data revealed directional flows corresponding to plant demand, carbon moving toward shaded saplings, nitrogen toward rapidly growing individuals. The network behaved dynamically, redistributing resources in response to environmental change. Molecular biology techniques deepened insight further. DNA sequencing allowed scientists to identify fungal species present in soil samples. Genetic markers traced which fungi connected which trees, mapping network topology across forest plots. Some fungal species form broad host connections, linking multiple tree species. Others specialized in specific partnerships, creating layered subnetworks within the larger system. Understanding these species relationships helped explain variations in resource flow and communication efficiency. Experimental manipulation confirmed functional roles. Researchers severed fungal connections between trees while leaving roots intact. Nutrient transfer declined sharply. Warning signal transmission slowed. Sapling survival rates dropped in shaded environments. Restoring fungal contact reinstated exchange patterns. The fungal web proved essential rather than incidental. Long-term ecological studies provided macro-scale confirmation. Forest plots monitored over decades revealed that trees connected through shared mycorrhizal networks exhibited synchronized growth patterns, stress responses, and nutrient cycles. Carbon allocation shifted seasonally across species lines. Drought resilience correlated with network density. Forest regeneration followed fungal connectivity patterns more closely than simple seed dispersal maps. These observations aligned with microscopic and isotopic evidence. Technological evolution accelerated discovery. Portable gas analyzers allowed real-time measurement of photosynthetic carbon flow. Soil respiration chambers tracked microbial and fungal metabolic activity. Remote sensing combined canopy imaging with ground-level nutrient mapping to correlate above-ground vitality with below-ground connectivity. Integration of these tools painted increasingly detailed portraits of forest networks. Yet acceptance of these findings was not immediate. Early claims of tree cooperation and communication met skepticism within scientific communities. The notion that trees shared resources challenged entrenched competitive models rooted in classical evolutionary theory. Some researchers argued that observed transfers were incidental rather than functional, passive leaks rather than adaptive exchanges, but accumulating evidence gradually dissolved resistance. Replication across ecosystems, boreal forests, temperate woodlands, tropical rainforests, demonstrated consistent patterns of nutrient and signal exchange mediated by fungal networks. Controlled experiments, field observations, and molecular analysis converged. The Wood Wide Web moved from hypothesis to accepted ecological principle. One of the most influential breakthroughs came from long-term isotope tracking studies led by forest ecologists studying carbon exchange between birch and fir trees. These studies documented bidirectional carbon flow varying seasonally, birch supporting fir in spring, fir supporting birch in autumn. The reciprocity reinforced the concept of interspecies cooperation mediated by fungal intermediaries. Scientific language began shifting accordingly, from competition models to network models. Technological refinement continues expanding understanding. Nanosensors capable of detecting minute chemical concentrations now monitor root exudates in real time. Advanced tomography imaging maps root and fungal structures in 3 dimensions without destructive sampling. Artificial intelligence assists in modeling nutrient flow patterns across complex network architectures. Each innovation reveals deeper layers of connectivity. Human discovery of these systems remains relatively recent, largely unfolding within the past half-century. For millennia, forests communicated, shared, warned, and adapted through underground networks unnoticed by human awareness. Only with modern instrumentation did the hidden architecture become visible. The realization reshaped forestry, conservation, and climate science. Reforestation strategies began incorporating fungal inoculation. Logging practices adjusted to preserve network hubs. Carbon cycling models expanded to include mycorrhizal redistribution dynamics. Understanding forests as interconnected systems transformed ecological management approaches. Even philosophical perspectives shifted. Trees, once seen as silent competitors, emerged as participants in cooperative, communicative networks sustained by fungal intermediaries and biochemical exchange. The forest floor, once dismissed as inert substrate, revealed itself as a living communication matrix. Standing today in a quiet woodland, it is possible to recognize that beneath the apparent stillness lies a vast system humanity only recently learned to perceive. Isotopes tracing invisible pathways, microsensors detecting silent electrical pulses, soil analysis revealing fungal webs binding species together. Science did not create these networks. It merely uncovered them, translating the slow language of roots and fungi into data, images, and measurable flow. A discovery not of something new, but of something ancient, finally brought into human understanding through the patient evolution of observation, technology, and curiosity. To stand at the edge of a forest is to feel the presence of something larger than its visible parts. At first, the mind counts individuals. One tree, then another. Birch beside pine, fir beside oak. Each trunk rises separately, each crown reaching for its own share of light. The eye interprets separation instinctively. Distinct organisms rooted in common soil but living independent lives. Yet the deeper science has revealed that this perception, though visually accurate, is functionally incomplete. Forests do not behave purely as collections of individuals. They behave as systems. And when examined through the lens of underground networks, chemical signaling, electrical communication, nutrient redistribution, and interspecies cooperation, the forest begins to resemble something far more unified, a vast breathing entity composed of many bodies, but sustained by shared function. The idea of a forest as a superorganism emerges from this realization. A superorganism is a biological system in which multiple organisms operate collectively as a single functional unit. Ant colonies, beehives, and coral reefs are often cited examples, individual members performing specialized roles that sustain the larger whole. Forests, though structurally different, exhibit parallel dynamics. Consider respiration. Individually, trees exchange gases through their leaves, absorbing carbon dioxide, releasing oxygen. Yet at the ecosystem level, these exchanges synchronize. Dense forests regulate atmospheric composition locally, influencing humidity, temperature, and even rainfall patterns. The forest breathes collectively. At dawn, stomata open across millions of leaves. At night, respiration continues through trunks and roots. Gas exchange pulses rhythmically across the landscape, a distributed respiratory system spanning kilometers. Water movement reinforces this unity. Through transpiration, trees draw groundwater upward and release it as vapor into the atmosphere. This vapor contributes to cloud formation and regional precipitation cycles. Forests effectively pump water from soil to sky, shaping climate patterns beyond their boundaries. In large systems like tropical rainforests, this hydrological function becomes so significant that deforestation alters continental rainfall regimes. The forest circulates water as a single organism might circulate blood. Nutrient exchange further dissolves individuality. Carbon produced in one tree flows into fungal networks and supports another. Nitrogen extracted in one soil patch nourishes distant roots. Minerals released through decomposition of a fallen trunk sustain saplings generations later. Energy moves continuously through shared pathways, unconstrained by species or genetic boundaries. The forest metabolizes collectively. Defense responses mirror immune systems. When insects attack one tree, chemical and electrical signals spread warnings across the network. Neighboring trees activate toxins. Predator-attracting volatiles release into the air. Fungal pathways transmit distress compounds underground. The forest mounts coordinated resistance. Like immune cells responding to localized infection, trees respond to shared threat signals, reinforcing defenses beyond the initial injury site. Memory integrates into this superorganismic identity. Seasonal stress, drought, frost, fire imprints physiological changes across entire forest communities. Growth rates synchronize. Bud burst timing adjusts collectively. Seed production fluctuates in coordinated pulses following environmental hardship. The forest remembers as a distributed archive of experience encoded across millions of organisms simultaneously. Regeneration reflects developmental continuity. When old trees die, nutrient redistribution feeds younger growth. Canopy gaps allow suppressed saplings to accelerate. Fungal networks guide resource flow toward regeneration zones. Death becomes renewal within the larger organism, tissue turnover rather than systemic loss. Even structural organization reveals systemic design. Root networks stabilize soil collectively, preventing erosion. Canopies regulate microclimates, buffering temperature extremes. Understory vegetation maintains humidity and nutrient cycling. Each layer contributes to overall system health. No single tree performs all functions. but together they sustain ecosystem equilibrium. Communication binds these processes. Chemical signals, electrical pulses, fungal mediation, airborne volatiles, all form channels through which information flows. Environmental detection in one region spreads awareness across the network. The forest senses collectively. It detects drought through root moisture gradients. It detects herbivory through chemical distress. It detects temperature shifts through cellular stress responses. Awareness disperses across living infrastructure. Time perspective deepens the superorganism analogy. Individual trees live decades or centuries. Forests persist millennia. Species composition shifts, yet fungal networks, soil microbiomes, and ecological functions continue across generations. The forest maintains identity despite turnover, like a body renewing cells while preserving form. Ancient forests exemplify this continuity. Some fungal networks in old-growth ecosystems span centuries, linking trees long dead to those still growing. Nutrients from past generations circulate into present growth. Ecological memory accumulates across time. The forest becomes history embodied. Human perception struggles with this scale. We are accustomed to organisms defined by skin boundaries, centralized organs, and rapid signaling systems. Forests operate through diffuse boundaries, decentralized control, and slow communication rhythms. Their unity emerges not from physical enclosure, but from functional integration. Climate interactions extend this integration outward. Forests influence atmospheric chemistry, carbon sequestration, and global temperature regulation. Through photosynthesis, they absorb carbon dioxide, mitigating greenhouse gas accumulation. Their superorganismic function extends to planetary metabolism. Disturbance reveals systemic fragility. Clear-cut deforestation does not merely remove individual trees. It collapses communication networks, disrupts hydrological cycles, destabilizes soil systems, and fractures nutrient exchange pathways. The superorganism suffers systemic trauma. Recovery requires reestablishing connectivity, fungal regrowth, soil restoration, species diversity return. Reforestation without network reconstruction often yields weakened ecosystems, lacking the integrated function of mature forests. Technology increasingly recognizes this unity. Conservation strategies prioritize preserving old-growth hubs, maintaining species diversity, and protecting fungal networks. Climate models incorporate forest system behavior rather than treating trees as isolated carbon units. Science moves toward ecosystem-level thinking. Philosophically, the superorganism concept reshapes humanity's relationship with forests. Rather than viewing them as resource collections, timber inventories, or carbon offsets, they appear as living entities with integrated physiology, communication, and adaptive capacity. Not conscious in the human sense, yet functionally alive at collective scale. Walking through such a forest becomes an encounter not with many lives, but with one vast life expressed through many forms. Each tree a cell, each fungal thread a nerve, each nutrient flow a circulatory current, each chemical signal a whisper in a distributed mind, breathing through leaves, drinking through roots, remembering through rings and genes, defending through toxins and alliances, renewing through decay and growth. The forest does not move as animals move. It does not think as humans think. Yet it responds, adapts, and sustains itself through coordinated processes spanning distances far beyond any single organism's reach. It lives slowly. Its conversations unfold over seasons. Its decisions manifest over decades. Its memory stretches centuries. To perceive it requires patience, a willingness to look beyond trunks and leaves into the invisible systems binding them together. And when seen in that light, the forest transforms from a gathering of individuals into a single, breathing presence, vast, cooperative, and enduring. Not many trees sharing space, but one living system sharing life. And so, beneath the quiet stillness of the forest, a vast living network continues its slow, patient conversation. Roots touching roots, fungi carrying whispers, trees sharing strength across generations. What appears silent is deeply alive. What seems separate is profoundly connected. The forest breathes, remembers, protects, and sustains itself as one great living system, ancient, cooperative, and endlessly calm. So as your thoughts begin to slow, imagine yourself resting beneath those towering canopies, the earth steady beneath you, the quiet language of trees moving softly below. Let the forest hold the noise of the world for a while, and allow your mind to grow still, your breathing gentle, your body at rest. Sleep peacefully.